Our next speaker is Sebastian Lourido. Sebastian is a Whitehead Fellow at the Institute next door. He will be telling us about conquering the genome of an apicomplexan parasite.
Thank you so much. I think this is a wonderful opportunity to see how these really transformative technologies can blur the lines between model and non-model organisms. And I'm really happy to participate in this session. I'll tell you about one of the projects that has been the focus of much of my career as a fellow, where the focus of the lab has really been on the basic biology of this group of protist parasites called the apicomplexans. A few notorious apicomplexan parasites include those that cause malaria, which remains a real scourge of the developing world, and cryptosporidiosis, which has recently been recognized as a major cause of infant mortality. Toxoplasmosis is perhaps less virulent, but much more prevalent, with nearly a quarter of the world's population being infected and causing threatening diseases in pregnant and immunocompromised individuals. And part of the fascination of studying the biology of these organisms, and also the challenge, is that their biology is quite distinct from that of other model organisms, which have typically been the focus of molecular biology. And so many of the lessons from yeast and mammalian cell molecular biology can't be readily translated to apicomplexan parasites, requiring us to study things more locally. Taking a closer look at the apicomplexan phylum, certain organisms from this phylum can be cultured in the lab. Several Plasmodium, malaria-causing organisms have been cultured, and more recently, Cryptosporidium parvum has been manipulated. But I'd like to emphasize that Toxoplasma gondii, amongst these organisms that can be cultured in the lab, has several experimental traits that really draw it apart from other apicomplexan organisms and that allow us to perform manipulations which mimic those that can be performed in other model organisms. In part, I'd like to focus on a couple key traits, a balanced GC content, which of course is advantageous for the identification of guide RNAs, and the ability to continuously culture Toxoplasma gondii in human host cells in the lab, which distinguishes it from many of the other organisms that can be genetically manipulated. And finally, the ability of Toxoplasma to repair double-stranded breaks through non-homologous end joining, which really is advantageous for the CRISPR-based genetic disruption strategies that I'll discuss in a minute. Just to take you briefly through the cell cycle of Toxoplasma gondii, it's divided into three major phases. It begins with the active invasion of host cells by these parasites. This is a trait that's common to many of the apicomplexan parasites. And then within those host cells, you have the creation of a replicative vacuole where these parasites can divide through several rounds prior to egressing from that cell to propagate further in other cells. And it's this cycle of invasion, multiplication, and reinvasion that really causes much of the pathology associated with apicomplexan infections. And so it has been our focus to study this stage of the parasitic life cycle, which can be completed in the lab in a matter of a couple of days on human fibroblasts. Many of the challenges that Toxoplasma undergoes during these transitions are common challenges to all of the apicomplexan phylum, and so our hope is to use these organisms to study the common biology of all apicomplexan parasites. As I started my lab as a fellow, we thought that perhaps CRISPR-Cas9 could be a useful tool to manipulate the genome of Toxoplasma gondii, and we adopted some of the early approaches that had been developed by the Sang lab and recoded them such that they could be expressed as a single plasmid system in Toxoplasma gondii, where the guide RNA was transcribed from the U6 promoter of Toxoplasma and the Cas9 nuclease from the same plasmid via a standard ubiquitous promoter. And that allowed us to generate double-stranded breaks in the toxoplasma genome, which upon transfection of this plasmid without any selection allowed for really unprecedented rates of gene disruption. Nearly 20% of the resulting vacuoles could be disrupted in this non-essential gene that's typically not lost in culture. And we could demonstrate that this high efficiency of gene disruption was mediated by non-homologous end joining, because parasites lacking a key component of that pathway were not able to disrupt genes with such efficiency, indicating that this pathway was performing the vast majority of these disruptions. So we thought that based on this efficiency, perhaps we could bridge the gap between genome-wide approaches, which had previously relied on genetic crosses and pre-existing variation, to gain the precision at a genome scale that targeted approaches had only been able to achieve on individual genes. And very much inspired by the work that had been done, in particular by our neighbor Tim Wang in the Lander and Sabatini labs at the Whitehead on genome scale screens using CRISPR-Cas9 on mammalian cells. However, we faced one key challenge, which was that Cas9 expression in toxoplasma was incredibly toxic. We could only achieve it in transient transfections, as I had shown you before. But we found a way around this by co-expressing Cas9 with what we call the decoy RNA, and a guide RNA that was not targeting any specific region of the toxoplasma genome. And the efficiency of the strategy is demonstrated here, where we express Cas9 with a fluorescent tag, either green or blue, in the presence or absence of a decoy guide RNA. And we show that by transfecting these separately and then mixing those populations, only the population that contains the guide RNA, the decoy RNA, survives over time and maintains Cas9 expression. And we show that this toxicity from Cas9 is dependent on the nuclease activity, because if you perform the same experiment with dead versions of Cas9, you see no shift between the blue and the green population. So toxicity is based on Cas9 activity, and we could overcome it by co-expressing Cas9 with a decoy guide RNA. With this in hand, we generated strains of toxoplasma that constitutively expressed Cas9, and we were able to achieve, by transfecting these parasites with a given guide RNA that could be integrated into the genome via selective marker, really outstanding efficiencies of gene disruption, almost arriving at 100% disruption of a given gene in the population. And this really set up the ability to perform pooled genetic screens in toxoplasma. And I'll walk you briefly through the strategy, because it mimics much of what has been done in mammalian cells. Cas9 expressing parasites can be transfected with a library of guide RNAs that spans the entire toxoplasma genome, about 10 guides for each of the 8,000 or so genes in the toxoplasma genome. And then this diverse pool of mutants is grown in human fibroblasts over several cycles to observe differences in the relative abundance of guide RNAs between the initial guide library population and then the final population. So for instance, guides targeting fitness-conferring genes, like the one shown in orange, would be lost from the population over time, whereas those that are targeting dispensable genes would be maintained in the population. And we can summarize the effects of these different guide RNAs as something we call a phenotype score, or others have called a CRISPR score, which is the mean log to fold change for guides against each gene. So one of the first scenarios we tried this strategy on was using a really standard assay for positive selection that relies on the ability of parasites to take up via salvage pathway a drug called FUDR, which intoxicates the pyrimidine pools in the parasites. And we've known for a while that knockouts in UPRT lead to resistance to these intoxicating agents because parasites can dispense with the salvage pathway and rely exclusively on the de novo synthesis pathway. So generating a diverse pool of mutants and selecting them, what we observed was a really dramatic enrichment of guide RNAs against UPRT and drawing on a phenotype score, UPRT above all other genes in the genome, and demonstrating the effectiveness of this strategy in identifying fitness-conferring genes under positive selective pressures. We also looked at our ability to identify fitness-conferring genes that are lost in the population through subsequent culture of these populations, and we were able to generate a ranking of all of the genes in the toxoplasma genome, shown here as bars on the 14 chromosomes of its genome and colored by their phenotype scores. If we zoom into a given section of this, we can then see, for instance, then that the pantothenate kinase is really important for parasite fitness under these conditions, and that, for instance, this ubiquitin ligase appears to be dispensable. And this gives us, for the first time, a complete map of the functional background for each of the genes in the toxoplasma genome, and really a roadmap for understanding the molecular biology of many of these genes with previously unknown functions. Just to give you a bit more confidence on our data set, we curated from the literature a list of known dispensable or essential genes that had been previously characterized in individual studies, and we saw a really beautiful sorting of the dispensable genes at the top of the rankings of phenotype scores and the essential genes at the bottom, with perhaps one exclusion, that of RAB4, which we were actually able to later knock out and demonstrate that it was a misannotation of the literature. Our phenotype scores also conformed with broad genomic trends such as expression, conservation, and gene ontology. And just to give you a small snippet of what that looks like, in ranking of genes by phenotype score, we saw an early collection of ribosomal proteins, for instance, which would be predicted to be essential at low phenotype scores, and these surface antigen genes, which are highly replicated and most likely dispensable in most cases, are collected much later, along with genes that are not expressed during the life stage that we're assaying, but expressed at a different stage called the bradyzoite stage. So all of our phenotype scores seem to be conforming to our expectations, and we took a closer look at another aspect of this biology of apicomplexans that is so difficult to study, which are those genes that have no known annotation. Another way of sorting the genes in the toxoplasma genome is by their depth of conservation. And with increased depth of conservation, we saw the expected increase in proportion of genes that are fitness conferring, with this highlighting a particular category of genes that was striking to us, which was those that are apicomplexan specific and fitness conferring, which we dubbed the indispensable conserved apicomplexan proteins. And in particular, we focused on those that had no annotations and no known domains. And so their function would be really difficult to otherwise examine. We decided to again use CRISPR to tag these genes and get some information about their localization. And for those that we were able to tag, we saw really diverse localizations throughout these highly polarized cells. Some of these are really emblematic of particular structures at the apical end or organellar structures that we know. And in particular, I'll tell you about a couple of stories that came out of this analysis. The first one was that we saw about half of these genes localizing to the mitochondrion, which we had previously considered a really conserved and sort of uninteresting from the perspective of apicomplexans organelle. But this really drew our attention to seeing it as a locus of perhaps apicomplexan-specific variability. And in particular, one of these genes really struck us because by structural prediction, it appeared to be related to the ATP synthase B subunit. And this was striking because unlike mammalian cells, where by sequence and by previous studies, we can identify all of the subunits of the ATP synthase, in apicomplexans, in malaria and toxoplasma and others, the stator subunits of the ATP synthase can't be identified by sequence-based searches. And so it had been a bit of a mystery as to what this whole complex looked like. And we tried to validate what we called the putative B subunit by performing immunoprecipitation reactions, which identified, along with this ICAP2 protein that we had identified in our screen, all of the other known subunits of the ATP synthase complex, in addition to several subunits that appear to be exclusively apicomplexan. And this really suggests that we can identify some of the missing biology of these organisms through such studies. In the last minute or so, I'll tell you about an apicomplexan-specific adaptation, which is that ability to invade host cells, which is so pivotal to their life cycle. And what we did in that case was to perform a secondary screen that used individual guide RNAs in an arrayed format to screen for their disruption of the invasive ability of these parasites. We identified one of the ICAPs, ICAP12, as important for this particular phenotype. And I'll summarize a lot of observations that we gathered and tell you that it seemed to be localized to secretory organelles, which are found in many apicomplexans. They're called micronemes. And it seemed to have structural similarity to mammalian claudins, which hold many of our cells together. And so we called it claudin-like apicomplexan microneme protein, or CLAMP. And one remarkable feature of it was that we found a single version of CLAMP in every single sequenced apicomplexan genome to date, which really was a remarkable conservation for a gene that might be involved in invasion. And we were later able to demonstrate its role in invasion by generating an inducible knockdown of this gene, where the knockdown parasites are shown in green. And the take-home message of this is that once the parasites are knocked down in this particular protein, they are unable to invade despite trying and trying to invade their hosts, their cognate host cells. And with that, I hope that I've given you a sense of what we're doing and really what CRISPR has done for us to make from Toxoplasma gondii a model apicomplexan parasite. And I'd like to thank Simon Siddick and Diego Huet in the lab, who pioneered many of these strategies, and Benedict Marcus, who has done some of the follow-up on the decoy guide RNA analyses. And I'd like to thank the contributions of Tim Wang, who really was the inspiration and answered many questions during the development of these screens, and Jacqueline Niles and Suresh Ganesan, who actually have demonstrated that CLAMP in malaria parasites is also extremely important for the fulfillment of that life cycle. And with that, I'll take any questions.